Hello students, welcome back to my channel. In this video, we are going to learn about DNA replication. What is DNA? What is the need for the DNA to be replicated and what is its purpose? What are the types of DNA replication and what are the steps involved in DNA replication? We can learn in detail one by one in this video. DNA replication. So we all know that DNA is a double helical structure having the nitrogenous bases adenine, thymine, guanine and cytosine with a sugar phosphate backbone. And we also know that adenine will always pair with thymine and guanine will always pair with cytosine. So in our channel we have one video. In that I have explained detail about the DNA structure. So for your reference you can refer that video. So what is DNA replication? DNA replication is nothing but a cell makes an identical copy of its own DNA. So in this picture you can see that the blue color represents the parental DNA and the red color represents the daughter DNA. So as we all know that where is DNA located? It is located inside the nucleus. So it is present in the form of chromosomes. So inside the nucleus it is tightly packed in the form of chromatin. So once the cell begins to divide, the first step that has to be taken place is the DNA should be replicated. Then only the cell enters into the cell division. So we have learned detail about the cell division. So I have also uploaded that video in my channel. You can go through that. What is cell division, how it is taking place in different stages. So during the cell division, in the interphase stage only the DNA replication takes place. So at this stage what happens, the chromatin loosens itself and it forms chromosomes, and the chromosomes again it loosens itself and gives the way for the replication of DNA that you can clearly see in this picture. Okay. Now what is the purpose of DNA replication? Why the DNA should be replicated? So it is essential for our cell growth. So before cell division the DNA has to be replicated, and then it is also involved in a repair or regeneration of the cells. And the last one is very important, that is in reproduction or inheritance. We say that inheritance, here the transmission of genetic information from parental strand to the daughter's DNA takes place in this. Okay. Now, what are the steps involved in DNA replication? The first step we call it as initiation. So, how does the replication begins? So, once the cell is ready for the cell division, during the interphase stage of the cell division, the DNA replication takes place. So, once the cell is ready, we can see that bubble formation in the chromosome. That is, it is unwinding, it is giving the signal that it is ready to replicate, and this bubble formation in the original position we call it as a replication bubble. So under the microscope we can see that, see chromosomes with the replication bubble, it indicates that the cell is ready for replication. Now so to replicate, the DNA has to unwind. So already we know that the base pairs adenine pairing with thymine and guanine pairing with cytosine. So in between the bases we have the hydrogen bonding. So that bond should be broken so that only DNA can replicate. So it takes place with the help of the enzyme DNA helicase. So the DNA helicase, it disturbs the hydrogen bonding between the base pairs. So the strands form a Y-shaped structure. So we call it as a replication fork. So this area will be acting as a template for replication to begin. So in the structure we can clearly see that replication fork, it is in Y-shaped structure, and this is the enzyme helicase which is helpful to unwind the DNA by breaking the hydrogen bond between the nitrogenous bases. See, once the replication is about to begin, we can see the replication bubble and the replication fork, and here the replication, it takes place in both directions. So it is bidirectional from 3' to 5' end as well as from 5' end to the 3' end. Now, so for the DNA to be replicated, it needs a primer. So what is a primer? So primer is a short piece of RNA which binds to the 3' end of the parental strand. So it is generated by the enzyme DNA primase, and it always binds at the starting point for the replication. In this picture you can clearly see the attachment of the primer. So this 3' end to the 5' end, you can see the forward primer attaching to the 3' end of the parental DNA, and in opposite side you can see 3' end only the primer get attached, and it is in a reverse order, so it is, we call it as the reverse primer. So now the primer is attached to the complementary strand of the parental DNA, and the next step is elongation. That means addition of nucleotides complementary to the parental strand. So in this figure you can clearly see the elongation process. Once the primer get attached to the 3' end of the parental strand, the enzyme DNA polymerase attaches with the parental strand and along with the primer. So it helps in the addition of nucleotides complementary to the parental strand. Okay, see it shows the direction of synthesis from 3' to 5' end, and down you can see from 5' to 3' end. Okay. Now, what is this DNA polymerase enzyme? So, this DNA polymerase enzyme is the main enzyme for the replication of DNA. So, we have two types of cells, prokaryotes and eukaryotes. So, in prokaryotes, DNA polymerase 3 is the main enzyme for replication, and DNA polymerase 1, 2, 4 and 5, they are used for error checking and repair of the DNA. As well as in the eukaryotes, we have polymerase alpha, delta and epsilon. These are the primary polymerases that are involved in DNA replication. So you can see here, as I already told, the DNA replication takes place in both the directions. So the replication from 3' end to 5' end, we call it as the leading strand, and from 5' end to the 3' end, we call it as the lagging strand. Okay, so in the leading strand, the replication of DNA is continuous, but in lagging strand, the replication of DNA is discontinuous, because it produces a pieces of DNA here and there, and at last they will be joined. So this picture clearly shows the formation of DNA sequences both in lagging and leading strand. So as I already told, from 3' to 5' end, the process we call as the leading strand, and from 5' to 3' we call it as the lagging strand. So in the lagging strand we can see multiple primers attached to the daughter DNA, and you can see here the pieces of the DNA. So it we call it as the Okazaki fragment. So once the replication is completed, the Okazaki fragments, that are the short pieces of the DNA, will be joined with the help of another enzyme to form a complete daughter DNA. Okay. So here helicase, we already learnt about the enzyme helicase. What is the function of the enzyme helicase? It helps in unwinding the DNA. It breaks the bonds between the nitrogenous bases, and this enzyme DNA polymerase helps in the elongation of the nucleotides complementary to the parental strand. And another enzyme we can see here is the topoisomerase. And what is the function of this enzyme? It helps in unwinding the DNA, and it prevents the supercoiling of the DNA. Okay. Now the last step is termination. So once it gets the signal the DNA is about to finish its replication, the enzyme exonuclease, it removes all the RNA primers. So in the insertion step, we learned that RNA primers get attached to the parental strand. No? So these RNA primers will be removed, and that place will be replaced with the appropriate bases. And then proofreading takes place to check, remove and replace any errors that have taken place in the DNA, that is daughter DNA. And as I already told, the Okazaki fragments will be joined with the help of the enzyme DNA ligase. And at last what we can see, parental and the daughter DNA, they will combine together to form a double helical structure of a DNA. Before the completion of the replication of DNA, there is another interesting thing, that is telomere. So what is this telomere and where it is present? So telomere are the short repeated sequence of the DNA that can be seen at the tip of the parental strand. So why it is present there? It acts as a protective cap, preventing the fusion of chromosome with the other chromosome. Okay, and this telomere should be replicated. So this telomere is replicated with the help of the enzyme telomerase. Once this replication is also over, the parent and the complementary strand, that is a daughter strand which is newly formed, coils into a familiar double helical shape of a DNA, and it shows that the replication is ended. So you can see here. So At the beginning, replication bubble will be formed and the replication fork will be also formed with the help of the enzyme helicase, and then elongation takes place with the help of the enzyme DNA polymerase. Once everything is over, we can see a new strand with the parental strand as well as the daughter strand. So during the elongation, we have two strands. One side it is called as the leading strand and other side we call it as the lagging strand, and Okazaki fragments can be seen in the lagging strand, and these fragments are joined together with the help of the enzyme DNA ligase. And finally we can see the replicated DNA. The green color shows the parental and this purple color shows the daughter DNA molecule. Okay. Now, what are the different types of DNA replication? There are three different types of DNA replication. The first one is conservative method. Here, one DNA molecule with both original strand and one will be the entirely new molecule. So, the parental strand and the daughter strand will be separated and they form the double helical structure of DNA in conservative method. Semi-conservative method means you can see two identical DNA molecules, each consisting of one strand will be the parental strand and another strand will be the daughter strand. And the last method is dispersive method. Here both new DNA molecules contain a mix of parental and the newly synthesized DNA distributed throughout both strands. So it is the fusion of both parental and the daughter DNA. This picture clearly shows the different types of DNA replication method. The first one is conservative method. So at the end we can say that, see here, the parental DNA will be separate and the new DNA will be separate. In semi-conservative method, one strand will be the parental strand and the other strand will be the daughter strand. And in dispersive method, you can see the mixture of that. So at the beginning you will have the parental strand, in between you will have the daughter strand of DNA. See? So this is the parental strand. So when the cell wants to divide, before cell division, what happens? DNA has to be replicated and the replication of the DNA takes place in the interphased stage of the mitosis. Again, you can see here, after every process is completed, initiation, elongation, you can see a complete DNA. Okay, so what is this method of DNA replication? Yes, it is semi-conservative method because you have one parental strand and one daughter strand forming a double helical structure. Do you understand? Thank you. Thank you for your listening and happy learning.